Hello, and welcome to the first installment of the DR Vision Voxel series. This will be the first webinar entitled Introduction to Data Visualization. We're going to be aiming this series at scientists with entry-level image analysis skills who are looking for a practical approach towards improving. My name is Willie, and I handle the technical and application support here at DR Vision, and I'll be your host today. Our goal in this webinar is to take a very practical approach to image and data visualization. We're going to take a look at some real fluorescence microscopy data, and we'll see some of the challenges in getting good visualizations of this data. I'm going to demonstrate this in our very own software called Aivia. And if you'd like to follow along, you can download a free 30-day trial from our website, drvtechnologies.com. While I will be demonstrating in Aivia, the concepts we present in this webinar are not exclusive to Aivia. They'll work with pretty much whatever software you do use. Also, we're hoping to keep this to a practical introduction course. So if I'm behaving myself, this won't be a math-intensive theory-first course. There are other great resources for you to find that kind of information. Today, we're going to start with some simple single-channel, single-frame 2D fluorescence microscopy data. Then we'll build on that to add more channels, time-lapse sequences, and even 3D images. Throughout this webinar, feel free to ask any questions you have that may come up. I have our sales and marketing manager, Quinn, here with me today, and she will be moderating the chat, as well as our Twitter, LinkedIn, and website. You can post any questions in any of these locations, and we'll do our best to answer them. All right, so let's dive right in. Before we load up our first data set, I'll give you a brief tour of our software interface. If you can see my cursor on the screen, we'll start up here in the left corner where we have a top menu for many basic interactions with the software. Below that, we have our View Controls toolbar, which allows us to manipulate how we view the data. This large section right here is the image panel, where we will see the loaded field of view as well as any objects we create later in this webinar series. Below that, we have a contents panel, which houses several modules for interacting easily with image data, though today we will be primarily concerned with this, the Image Explorer. On the right side, we have this side panel, and we'll be focusing on this sub-panel in the top right corner that houses our channels and their associated parameters. Now we can start by loading some data. Simply go to File, Open, and select the data we'd like to work with. Here I've selected a membrane TIFF image. There are many kinds of file formats out there, and they all have their advantages. You are probably familiar with common ones like JPEGs, PNGs, or even bitmaps. One popular format that we will be working with today is the tagged image file format, or .tiff. We'll see some of the reasons it is popular as we go through this webinar. One of the first things we'll see is a little pop-up dialog asking us to calibrate the image. When data is displayed on the screen of a computer, it probably only knows the dimensions of the image in terms of pixels and brightness or intensity values. But we can tell the software how to calculate real, meaningful measurements in relatable units with this calibration. For this image, I know that the X and Y dimensions are at 0.10833333, so on micrometers per pixel. So we can apply that. If we want to adjust this later, we simply go down to this little pencil in the bottom left corner and we reopen our calibration properties dialog. But we'll leave it as it is for now. The first thing we see when we load this data is it's a pretty dark image. We can't just give up here, however, so we'll start by adjusting the levels in the lookup tables to increase the visual contrast. In our channel panel, we drag these sliders, moving the slider on the right towards the left, helps make the bright parts seem even brighter, and dragging the slider on the right, and dragging the slider on the left towards the right, makes the dark parts even darker. As we adjust this, we can find the perfect balance. Now, a slider is great, it's easy to use, but if we want to quantify this process a little bit more, we can click the arrow next to the channel name. Now we see a distribution of all of the intensities in the image, as well as numerical values for the sliders. For this image, I know that some good values are 1000 to 4500. Look at that. Now we can clearly see the data in this image. What we have here are some cell membranes. We can zoom in on this image, even pan around to see different areas of the field of view. If we zoom in far enough, we can even see individual pixels, or voxels for three-dimensional images. Some of these pixels don't look exactly right, do they? That's what we call noise. Every image will have some noise, and here it looks like we have some low intensity background fluorescence causing a noise. A good image, however, will have a lot more useful data in the signal than noise, and this is often called signal to noise ratio, or SNR. A high SNR image typically makes a better looking image. They are easier to process and more reliable to analyze. Lastly, let's talk about the color in this image. The microscope, when taking this, didn't necessarily capture red wavelengths, even though the image here appears red. It probably just recorded the intensity signal. This color here is artificially added by the software, and we can change it to any color we'd like. For example, a very pretty fuchsia, or a bright cyan. This is helpful because we can add another channel to get more information out of this image. To load another image, we can just go to File, Open, and this time I'm selecting Dual Channel. We can already sort of see the membranes, but there is another channel that takes other information from the same XYZ location in the same time frame. We can adjust the levels in this image just in the same way we do for a single channel image. We'll start with the membrane channel again. It's a slightly different number here. I like 16 to 180 for these values. Here we see our bright red looking very good. However, this blue channel, you can hardly see it. So we're going to adjust this from 0 to 5. Now we can clearly see the value of the lookup table, as it allows us to select the images that most closely, or the values that let us most closely see what we want to in the image. It is important to note that these two channels, though they are from the same space and time, act somewhat independently in the software. So we can adjust the overall visualization without affecting the membrane channel while we change this DNA calibrated channel. Now you may be wondering, why are these channels so different from each other? Different levels are necessary because the data collected has different properties. Different properties such as different physical structures, like you can see here, or different fluorophores used in imaging. There are numerous reasons why you might want to visualize multiple channels. Common reasons are to see different physical structures at the same time, such as in this dataset, or perhaps to visualize the co-localization of different fluorescent labels. It is still worth keeping in mind that the colors we have displayed here are artificially applied by the lookup table that we have selected. They don't necessarily represent the reality of the fluorescent label used. We can change this again. If perhaps we don't like blue, we want to go to this pinkish color here, or bright green. Now, while there are multiple color channels in this image, they are in the same space and taken at the same time frame. But we can extend this to have a time dimension in image visualization as well. Let's begin by constructing our own time-lapse image. I'm going to go ahead and clear these so we have a nice empty workspace to work with. In Aivia, we might have a series of images all taken of the same cells in the same field of view, but at different time points. We can load these by going to File, Import, Image Sequence. A little dialog pops up that's going to help guide us. We can load all the frames by selecting Load Image and selecting all of the images in our sequence. As we have loaded these, Aivia tries to automatically assign these images to a sequence. We can see that it reads the file names and assigns them a place in our timeline, automatically ready to go. We just need to set the name. We'll call this time-lapse. Once again, we get the option to calibrate. Since we're not actually taking any measurements here, we can feel free to leave this image and some of the subsequent images uncalibrated. Once again, we have to begin by adjusting our lookup table. For this image, 100 to 1000 is a pretty good range to start with. We can easily see all of the cells in this field of view. If we look below this main image panel, we now see a time-lapse bar. We can step through each frame or play the sequence as a whole. This slider to the right here will help us adjust the playback speed. We can decrease it and make it playback slower, or increase it and get the fastest playthrough possible. Now that we have begun to play through this image, we can see how these cells are moving around. In the future, we'll talk about how to count how many cells are in an image like this, or be able to automatically track their movements. The playback speed we have set here is not necessarily the real-time speed of the cells in this image. Just like with spatial dimensions, x, y, even z, when dealing with time-lapse images, there is a time resolution that describes the elapsed time between consecutive frames. Here we have left it uncalibrated and artificially set the playback speed significantly higher than real life, as these cells tend to move very, very slowly. All of the principles and adjustments we have made to single frame images so far in this webinar can also be made to time-lapse images, though there is often a trade-off. With so many images in a time-lapse sequence, it may not be feasible or even possible to accurately adjust every single frame so that the parameters are perfect. The trade-off we have to make is to generally select parameters that look good applied to the entire sequence. Now that we have stitched all of these images and worked hard to find just the right adjustments, we may want to share this sequence and make that process much easier for others. In Aivia, we can select File, Export, Movie, and quickly save this movie or the sequence to one of several popular movie formats. These include things like AVI movies, MP4s, QuickTime, or Windows Media videos. This is useful for more generic, common video players. However, one of the advantages I mentioned before of the .tiff file format is the ability to save a time-lapse sequence like this in a single file. We can just go to File, Save, and save our image as a single file. I've already saved this image, so let's see what it looks like when we open it. Again, we've left this image uncalibrated. Once we adjust our levels again, we can play through without having to reconstruct the entire sequence from individual frames. So far, we have dealt with only two-dimensional images, but this idea of a time-lapse sequence can also be used to create 3D representations of images with a z-dimension stack, as opposed to a time dimension stack. Let's clear our workspace once again. And just like with loading a time-lapse sequence, we can import each z-slice to build a 3D reconstruction. Once we've selected all of the images, again, Aivia tries to assign a time-lapse sequence. However, we know that that's not correct in this case. So we'll switch from automatic file naming to manual file naming. We know that this dimension should be in Z rather than in time. Once we change that, Aivia automatically readjusts all of our images to import them into the correct Z location. Once again, we can name our file and we're good to go. This image we've also left uncalibrated, as we're not taking any specific measurements, so we don't need to worry about whether the computer views it as just a pixel or measures it in micrometers, nanometers, or millimeters. Now, once we've loaded this, we can step through it, slice by slice, even play through the entire sequence. Even set at the highest speeds, we can see the neuron in this image sort of begin to take shape. However, playing through it slice by slice isn't exactly what we want now, is it? Sometimes 2D isn't good enough, especially when it comes to representing complex shapes, structures, or geometries. Now, fortunately, we can easily render this in 3D right in Aivia. We go up to our View Controls toolbar. So far, we have viewed everything in the main 2D view. But by clicking the 3D view, we can easily render this neuron. And here it is, clear as day. There are some additional features to be considered when viewing images in 3D. Features beyond what we have discussed in 2D so far. The most interesting of these is perhaps the different rendering modes, which is how we interpret data from each slice in a Z-stack. Here we have started off with a volume rendering. This interprets each bright intensity as the volume of the cell. We can also render as a solid surface. This tries to find the surfaces defined by the intensity in the cell, but the image itself in 3D remains hollow on the inside. This is great for a number of things, such as looking at the texture or potential texture of a cell or some sort of membrane. Lastly, we can also render as a wireframe image. This is something you might expect to see in 3D modeling. Now, what you're going to see here isn't all that great when viewed from a distance. It sort of blends in and just looks very flat. However, once we zoom in, we can easily see the individual texture of this dendrite here begin to take shape. Let's go back to our preview surface. We can rotate, zoom, and pan around the image to get a very clear understanding of the complex geometry of this neuron. This looks much better than just a simple stack of images played in a sequence. We can also adjust the ISO levels in a 3D image like this. By decreasing this, we're selecting more of the low value, low intensity pixels, voxels, in the 2D construction to include in our 3D reconstruction. Right now, we have also included the most bright intensity voxels from our 2D construction into our 3D reconstruction. But we can lower that if there were bright spots in the noise, or bright noise spots in the image. Now, one of the advantages of a TIFF, like we've talked about before, is that we can save an image in 3D just like this. We don't have to import each slice from a stack individually. Simply go to File, Save, and we can save this image just like we did before. I've already saved this, so I'll show you what it looks like once you've reloaded the image. Here we have our TIFF image opened, left uncalibrated, and we can clearly see this whole stack is reconstructed. And once we load it in 3D, we have our same image. Now you notice the color changed here, just like we've seen in other 2D and time-lapse reconstructions. We can artificially apply whatever color we choose. This is a very pretty green, or perhaps blue. One second. This has been the very basic introduction to visualizing images in 3D time-lapse, 2 dimensions with one or perhaps multiple channels. So thank you all for tuning in to the very first Voxel series webinar from DR Vision. I hope this information has been useful, and I hope you've learned a lot. I would encourage you to try out visualizing some basic data sets, and want to remind you that you can get a free trial of Aivia to practice with. Also, tune in for our next installment in the Voxel series, where we plan on looking at some basic analysis techniques. If you have any questions or topics you would like us to cover in this series, please feel free to reach out and let us know. We are excited to bring you the information that you would find the most helpful and informative. Before we sign off today, Quinn, are there any questions?
No, Willie, there are not.
All right. Thank you all once again for tuning in.